Number 1. Walliserops. Picture yourself crawling along a tropical Devonian seafloor 390 million years ago. The sand is warm. The water above you glows turquoise in the equatorial sun, and on the ocean floor in front of you, something is moving. It is small, only about 5 centimeters long, segmented, armored, and trilobite-shaped. But growing forward from its head is a 3-pronged trident. Not a horn, not a tusk, a literal fork. This is Walliserops. The genus was erected in 2003 by paleontologists Richard Forte and Brian Chatterton. They worked from material recovered at Jebel Issoumor in the Anti-Atlas Mountains of southeastern Morocco. The trident itself extends roughly 30 millimeters forward of the head shield. Its 3 parallel prongs are made of fused exoskeleton growing out of the central forehead structure called the glabella. For 2 decades, no one knew what the trident was for. Defense was proposed. So was sediment shoveling. So was prey detection. So was simple ornament. None of these explanations could be tested directly because trilobites have been extinct for 250 million years. Then, in 2023, Alan Gishlick and Richard Forte published a definitive paper in the Proceedings of the National Academy of Sciences. They compared the geometry of Walliserops tridents to the horns of modern beetles. Statistical morphometric analysis showed that Walliserops fell within the geometric range of weapons used in male versus male combat among rhinoceros and stag beetles. One specimen had a malformed, 4-pronged trident. This pattern matches the deformed horns of modern beetles whose larvae were injured during development. The conclusion was extraordinary. Walliserops became the earliest known animal to have evolved horn-like sexual weaponry. The origin of intraspecific male combat structures was pushed back nearly 400 million years. Walliserops belongs to the order Phacopida, family Acastidae. Its eyes were the schizochroal compound type, with hundreds of large lenses. They provided roughly 360 degrees of horizontal vision. The Sahara region during the Devonian was a shallow tropical platform. The waters were warm, they were well oxygenated. They supported dense trilobite faunas alongside early ammonoids and brachiopods. Walliserops shared its habitat with the related Urbinochile and the long-spined Cyphaspis. Not every paleontologist accepts the sexual weapon interpretation. Bridget Schoenemann has argued the prongs primarily deterred predators by making the trilobite appear larger. The debate continues. Walliserops itself disappeared during the late Devonian Kellwasser event around 372 million years ago, one of the big 5 mass extinctions. But its trident-bearing skull endures in museum drawers, evidence that the strangest experiments in animal architecture began far earlier than anyone suspected. And the experiment continues. Number 2. Yi Qi. In 2015, Chinese paleontologist Xing Xu and colleagues published a Nature paper that changed the understanding of dinosaur flight. The specimen was catalogued STM31-2 at the Shandong Tianyu Museum of Nature. It came from the Tiaojishan Formation in Hebei Province. The species name, Yi Qi, means strange wing in Mandarin. It is the shortest valid binomial name in zoological nomenclature. Yi Qi lived 160 million years ago. During the Middle to Late Jurassic, it was approximately the size of a large pigeon. Its mass was around 380 grams. Its body length, including the tail, reached about 60 centimeters. The defining feature is a long rod-shaped bone projecting from the wrist. Paleontologists call it the styloform element. It has been found in no other dinosaur. The styloform supported a membranous wing, similar in construction to those of modern bats and pterosaurs. Preserved patches of soft tissue remain around the bone, leaving no doubt about the membrane's existence. Yi Qi belongs to the family Scansoriopterigidae. This is a small group of arboreal theropods that includes Epidexipteryx and Scansoriopteryx. The fossil is doubly remarkable. The body was covered in feathers. The wings were leathery membrane. Carbon-rich fossilized melanosomes confirm the feathered body coloration. The wings show no sign of feathers. Yi Qi is the only known dinosaur with both feathered and membranous body coverings in distinct, non-overlapping zones. Whether Yi Qi could fly remains contested. In 2020, biomechanist Thomas Dissecki published an analysis concluding that Yi Qi was an inefficient glider. It almost certainly could not flap. A follow-up paper by Alexander Dissecki in 2022 reconstructed possible flight surfaces. He suggested Yi Qi could have launched only from elevated perches. Ground launching was impossible. The Taojishan ecosystem was a humid temperate forest dominated by ginkgophytes and ferns. It shared habitat with feathered theropods like Anchiornis and with early mammals. Yi Qi rewrote the understanding of dinosaur flight evolution. It represents a parallel, dead-end lineage that experimented with membrane wings independently of the line leading to birds. A second Scansoriopter rigid, Ambopteryx longibrachium, was described in 2019 from a different formation. This confirmed the membrane wing strategy was used by multiple species, not a single oddity. Both Yi Qi and Ambopteryx left no descendants. The feathered wing lineage that became birds outcompeted them. The membrane wings would have severely limited maneuverability compared to the precision and adjustability of feathered wings. This may explain why bats waited 50 million more years after Yi Qi to evolve a similar but more refined membrane structure. The discovery was named one of Time magazine's top scientific discoveries of 2015. It stands as one of the strongest examples of convergent evolution in vertebrates. Bat wings appeared on a dinosaur 50 million years before bats themselves existed, and then they vanished. But Yi Qi was not the only Mesozoic creature whose body looked impossible. Many of evolution's strangest experiments survived for far longer. Number 3. Diplocaulus. Travel forward in time into the swamps of Permian, Texas, around 280 million years ago. The water is shallow, the mud is thick, and lying motionless on the bottom is something that looks like a salamander wearing an anvil for a head. The skull is shaped like a giant boomerang. Its head is 3 times wider than long. This is Diplocaulus. The genus was first described in 1877 by Edward Drinker Cope. It came from the late Carboniferous through late Permian, 306 to 255 million years ago. Adult body length reaches about 1 meter. The head accounts for nearly 30 centimeters of total skull width. 2 cranial bones, the squamosal and the tabular, project sideways and backward. They form the boomerang silhouette as the animal grows. Juveniles do not look this way. Their heads are relatively normal and lizard-like. The horns develop progressively through ontogeny. They become exaggerated only in mature adults. Most fossils have been recovered from the Permian red beds of Texas, Oklahoma, and New Mexico. Hundreds of specimens are known. Diplocaulus is one of the best represented Permian amphibians in museum collections. The function of the head shape has generated multiple competing hypotheses across more than a century of research. The hydrofoil hypothesis was proposed by Jenny Cruikshank in 1990. It was elaborated by John Bolt and Hans-Dieter Suess. They argue the head provided lift while swimming, allowing rapid upward escape from predators. Computational paleobiologist Stephanie Pierce later supported this with finite element modeling. The biomechanics work. The defensive hypothesis suggests the wide head made it impossible for predators to swallow Diplocaulus headfirst. A predator's mouth simply could not accommodate the boomerang. The display hypothesis proposes the horns were sexual ornaments. The largest horns indicated reproductive fitness. The eyes were positioned on top of the head. This suggests an ambush predator that lay on the bottom of streams and ponds, looking upward. Diet consisted of small fish and aquatic invertebrates, captured by sudden upward strikes from the riverbed. A close relative, Diplocaulus, lived alongside Diplocaulus. It possessed an even more exaggerated skull shape. Diplocaulus survived through the Permian, but it went extinct during the Permian-Triassic extinction event 252 million years ago. This was the most severe mass extinction in Earth's history. Up to 90% of marine species disappeared. The animal would have lived in seasonal floodplain rivers and ponds. These periodically dried out. Diplocaulus may have been capable of estivation, surviving dry periods buried in the mud. Modern analogs are difficult to find. No living amphibian has comparable skull morphology. The boomerang head appears to have been a one-time experiment. Diplocaulus has appeared in popular paleontology since the 1930s, including in early Disney natural history programs and the BBC's 1985 documentary series. But Diplocaulus is far from the strangest creature evolution has produced. Even the Burgess Shale, 500 million years older, holds bodies that defy classification. Number 4. Hallucigenia. The Burgess Shale is a black mudstone formation in British Columbia, Canada. It was discovered by American paleontologist Charles Walcott in 1909. Walcott collected thousands of fossils. Most were stored in drawers at the Smithsonian, unstudied. In 1977, Cambridge paleontologist Simon Conway Morris began re-examining one of these specimens. He was looking at a worm-shaped creature about 30 millimeters long. It had 7 pairs of stiff spines on what Conway Morris took to be its underside. It had 7 pairs of soft tentacles waving from what he took to be its back. He named the genus Hallucigenia for its hallucinogenic appearance. The reconstruction was wrong. In 1991, Lars Ramskjöld and Hao Jiangwang published a re-evaluation based on Chinese specimens of related animals from the Chengjiang biota. The body was upside down. The spines were actually dorsal defenses. The tentacles were actually leg pairs. Even after the flip, paleontologists still got the head and tail backwards. They mistook the actual head for a gas bubble formed during decomposition. The error persisted for another 24 years. In 2015, Martin Smith and Jean Bernard Caron published a Nature paper that finally identified the head. They distinguished the head from the gas bubble. They identified a circular mouth with a ring of small radial teeth. They identified pharyngeal teeth lining the throat. Hallucigenia is now classified as a stem group Lobopodian. It is an evolutionary cousin of modern velvet worms in the phylum Onychophora. Modern velvet worms still possess soft, fleshy, unjointed legs analogous to Hallucigenia's tentacle legs. They live in tropical leaf litter today. They squirt sticky slime to capture insects. The dorsal spines of Hallucigenia are composed of organic phosphatic material. The structure resembles fingernails. Specimens are known from the Burgess Shale, the Maotinshan Shale in Yunnan Province, and the Sirius Passet biota in Greenland. Hallucigenia represents one of the iconic creatures of the Cambrian explosion. This was a 5 million year window beginning roughly 540 million years ago, during which most modern animal phyla first appeared. Stephen Jay Gould featured Hallucigenia prominently in his 1989 book Wonderful Life. He used it to argue that early animal evolution produced experimental body plans that later disappeared. The reinterpretation by Smith and Caron required scanning electron microscopy. It required high-resolution photography of dozens of specimens. They had to identify pharyngeal teeth that were less than a millimeter across. The radial teeth indicate Hallucigenia was a predator or scavenger, sucking in soft tissue. Pharyngeal teeth are also present in modern arthropods like priapulid worms. This suggests a deep evolutionary origin for this feeding apparatus. Hallucigenia became extinct by the end of the Cambrian period, around 490 million years ago. It left no direct descendants but contributed to the ancestry of velvet worms. But Hallucigenia was only the second strangest creature in the Burgess Shale. The strangest was kept in storage even longer. Number 5. Opabinia. Charles Walcott described Opabinia regalis in 1912. He classified it as a crustacean arthropod. The species name combines a reference to nearby Mount Opabin with regalis, meaning royal. For 60 years, Opabinia sat in collections, regarded as a slightly unusual early arthropod. In 1972, Conway Morris and his colleague Harry Whittington presented a re-examination at the annual meeting of the Paleontological Association. The audience reportedly burst into laughter at the reconstruction. They believed it was a joke. Whittington had to present his evidence in detail before they accepted he was serious. What Whittington showed them was an animal with 5 mushroom-shaped compound eyes mounted on stalks atop the head. No other animal in Earth's history has this configuration. He showed them a long, flexible proboscis projecting forward from the head. The proboscis ended in a circular grasping claw with backward curved spines. He showed them 11 pairs of lateral side flaps fringed with gill-like filaments. These were used for both swimming and respiration. He showed them a 3-lobed tail at the rear, used for steering and stability. The proboscis is the most striking feature. It would have functioned like an elephant trunk crossed with a vacuum cleaner. It picked up soft prey from the seafloor. It passed prey backward to a circular ventral mouth on the underside of the head. Opabinia is now classified as a stem group arthropod. It sits on the lineage leading toward modern arthropods but predates their key innovations. It is closely related to Anomalocaris, the famous Cambrian apex predator. Both share the tendency for unusual head appendages. In 2022, paleontologists Joseph Moyshuk and Jean Bernard Caron published a reinterpretation of Opabinia. They used new high-resolution imaging to identify finer sensory bristles along the proboscis. They argued the animal probed sediment for hidden prey rather than capturing free-swimming targets. Specimens are known almost exclusively from the Burgess Shale. A few rare examples have been found in the Spence Shale of Utah and the Maotinshan Shale in China. Opabinia lived around 508 million years ago in the Middle Cambrian. The first published reconstruction by Walcott in 1912 misinterpreted the proboscis as a feather-shaped tail. He thought the eyes were gills. In 2023, paleontologist Alison Daly published a morphometric analysis of the eye placement. She concluded the eye geometry provided overlapping panoramic vision similar to modern dragonflies. Opabinia was a visually oriented predator with depth perception. The five-eye configuration disappeared from the fossil record after the Middle Cambrian. No modern animal has retained anything similar. Opabinia contributed genetic ancestry to the modern arthropod phylum. It left no specific descendants we can identify. The body plan disappeared because true arthropods, with their jointed limbs and external skeletons, were more efficient. Opabinia represents an evolutionary draft abandoned in favor of better designs. But evolution kept producing strange anatomies long after the Cambrian closed. The Triassic period would deliver one of the most exaggerated body plans ever found. Number 6. Tanystropheus. The Triassic period followed the Permian extinction. Life had been devastated. Marine ecosystems were rebuilding from near extinction levels. In Switzerland's Monte San Giorgio region, the rebuilding produced something extraordinary. In 1852, paleontologist Hermann von Meyer described a creature with bones like nothing he had ever seen. They were elongated. They were thin. They were so unusual that von Meyer initially identified them as the wing finger bones of a giant pterosaur. The misidentification persisted for decades. In 1886, Italian paleontologist Francesco Bassani recognized the bones as cervical vertebrae. They belonged to a long-necked reptile, not a flying creature. The animal was named Tanystropheus. Tanystropheus lived in the Middle Triassic, 247 to 235 million years ago. Adult body length reached up to 6 meters. The neck accounted for approximately 3 meters. Half the total length was neck. The neck contained only 13 cervical vertebrae. Each was more than 30 centimeters long. Despite this dramatic length, the neck was relatively rigid. The proportions of the vertebrae and limited flexibility between them prevented free movement. Tanystropheus belongs to the family Tanystrophiidae. This is an extinct group of archosauromorphs. They are related to the lineage that produced dinosaurs and modern crocodiles. The Monte San Giorgio specimens are preserved in lake sediments. They are extraordinarily detailed. Some retain gut contents, allowing direct analysis of diet. Tanystropheus is preserved in 2 distinct ecomorphs. A small species, Tanystropheus longobardicus, has a 1.5 meter body. A larger species, Tanystropheus hydroides, reaches up to 6 meters. In 2020, paleontologist Stefan Speakman published a current biology paper using CT scans. The scans confirmed the small and large forms were distinct species rather than juvenile and adult of the same. Stomach contents in some specimens contain fish bones and cephalopod hooklets. This indicates a piscivorous diet. The next primary function appears to have been a fishing strategy. The body remained on land or in shallow water. The head darted out into deeper water to catch prey. The body itself was poorly suited for swimming. The tail is short relative to body length. Some paleontologists, notably Olivier Riepel, have argued Tanystropheus was fully aquatic. Others, including Speakman, favor a semi-aquatic lifestyle. The skull is small, relative to body size, with sharp, interlocking conical teeth typical of fish eaters. The Triassic Tethys ocean coastline was a complex of shallow lagoons and atolls. Tanystropheus shared its environment with nothosaurs, placodonts, and early ichthyosaurs. These were all members of the recovering Triassic marine fauna. Tanystropheus survived until the Carnian pluvial episode. This was a global wet phase approximately 234 million years ago. The episode reorganized Triassic ecosystems and ended Tanystropheus's lineage. Tanystropheus is one of the most extreme examples of disproportionate body proportions in any tetrapod. The neck-to-body ratio of 3:1 has never been matched, before or since, but the strangest mouths in evolutionary history were still to come. The Permian had already produced something that would not be solved for over a century. Number 7. Helicoprion. Russian geologist Alexander Karpinski was working in the Ural Mountains in the late 1890s. He found something unlike any fossil ever recorded. It was a tightly coiled spiral of teeth, about 30 centimeters in diameter. The teeth were sharp, triangular, and serrated. They were arranged in a continuous helix. There were 180 teeth in the spiral. They had been added one at a time, with new teeth growing from the inside as the animal aged. Karpinski published the description in 1899. He named the genus Helicoprion. The species name means spiral saw. The Helicoprion lived in the early Permian, 290 to 250 million years ago. But for over a century after Karpinski's discovery, no one knew where on the animal's body the whorl belonged. Karpinski himself initially placed the whorl on the upper jaw, curling outward like a forehead horn. Later, researchers proposed it was on the lower jaw. Others argued it was on the snout. Some suggested it was the dorsal fin, some said it was the tail. Some thought it grew inside the throat. Every imaginable position was proposed at some point. The breakthrough came in 2013. Leif Tapanila and Jesse Pruitt at Idaho State University used CT scanning on an exceptionally preserved specimen. The specimen was catalogued IMNH 37899. The scan revealed something no previous reconstruction had captured. The whorl occupied the entire lower jaw. There were no other teeth in the lower jaw. The upper jaw was completely toothless. Instead of teeth, the upper jaw bore a hard plate. As the mouth closed, the lower jaw whorl rotated against this plate. The action was a slicing motion, not a crushing one. The geometry was elegant in retrospect. Helicoprion is now classified within the order Eugeneodontida. This is a group of cartilaginous fish related to modern chimeras and silver sharks. They are not true sharks. Adult body length is estimated at 3 to 12 meters. The largest specimens were among the largest Permian marine predators. Diet was likely soft-bodied prey. Ammonoids and squid-like cephalopods were abundant in Permian seas. The slicing teeth were not designed for hard prey. The teeth themselves are flattened. Their edges are sharp and serrated. They were designed for cutting through soft tissue, not for piercing armored prey. Helicoprion fossils have been found in Russia, Idaho, Mexico, Australia, China, and Iran. The animal had a global tropical distribution. The largest known whorl, from the Phosphoria Formation of Idaho, is 55 centimeters in diameter. Helicoprion's cartilaginous skeleton means almost the entire body has been lost to fossilization. Only the heavily mineralized teeth survive. The teeth grew continuously throughout life. The spiral diameter directly correlates with age. The genus survived the end Permian extinction approximately 252 million years ago. It persisted briefly into the early Triassic before going extinct, leaving no descendants. But the most extreme weapon in evolutionary history was still ahead. It would not appear until the very end of the dinosaur era. Number 8. Therizinosaurus. In 1954, Soviet paleontologist Evgeny Maliev described claw bones recovered from the Nemec Formation of Mongolia. The claws were enormous. They measured up to 1 meter along the curve. They were the largest claws ever found in the fossil record. Maliev had no skeleton to work from. He had only the claws. He interpreted them as belonging to a giant turtle-like reptile. He named the genus Therizinosaurus, meaning scythe lizard. The interpretation was wrong. Subsequent discoveries revealed Therizinosaurus was a theropod dinosaur. It was not a turtle relative. It belonged to the family Therizinosauridae. This is a clade closely related to oviraptorosaurs and to birds. Therizinosaurus lived in the late Cretaceous, approximately 70 million years ago. It existed in the Maastrichtian stage, immediately preceding the Cretaceous-Paleogene extinction. Adult body length reached up to 10 meters. Body mass was 3 to 5 tons. Each manual claw measured up to 100 centimeters along the curve. They were the longest claws of any animal in Earth's history. Despite being a theropod, Therizinosaurus was an obligate herbivore. It had a small toothless beak. Behind the beak were leaf-shaped serrated cheek teeth. The pot-bellied body shape is typical of large herbivorous dinosaurs. It accommodated an extensive fermentation gut for processing tough plant material. Modern reconstructions show Therizinosaurus with extensive feathered plumage. This is based on fossils of related species like Bipiosaurus, which preserves complete plumage. The function of the claws has been debated for 70 years. Defense against predators is one hypothesis. Intraspecific combat is another. Foliage stripping has been proposed, so has digging for termites or roots. In 2018, Stefan Lautenschlager published a finite element analysis of Therizinosaurus claws. He concluded the claws were too gracile for digging or combat. The most likely function was hooking and pulling foliage toward the mouth. The claws were leaf pickers, not weapons. The Nemec Formation of Mongolia represents a humid floodplain ecosystem. The environment had rivers and conifer forests. Therizinosaurus shared its environment with the predator Tarbosaurus, the duck-billed Saurolophus, and the Ankylosaur tarchia. The skull of Therizinosaurus has never been found. Despite multiple complete forelimb specimens, no cranial material has been recovered. Closely related Bipiosaurus from the Yishan Formation of China preserves complete plumage. This confirms feathering across the entire family. Therizinosaurus is one of the most prominently featured dinosaurs in the 2022 Jurassic World Dominion film. It was depicted with unusual accuracy for a Hollywood production. Mongolian field expeditions led by Rinchen Barsbold during the 1970s produced most of the known Therizinosaurus material. The Therizinosaur lineage probably evolved from small carnivorous theropods. They gradually shifted to herbivory during the early Cretaceous. The shift involved enlarging the gut, modifying teeth, and growing the claws to extreme proportions. Therizinosaurus went extinct at the Cretaceous-Paleogene boundary alongside all non-avian dinosaurs. 66 million years ago, an asteroid impact and the resulting environmental collapse ended the lineage forever. But strange anatomies in prehistory often came in pairs. The Devonian seas had produced something even more extreme. Number 9. Dunkleosteus. The late Devonian was an age of armored fish. 382 to 358 million years ago, the seas were dominated by placoderms. Placoderms had bony plates fused around their heads and pectoral regions. They are completely extinct today. They left no descendants. The largest placoderm was Dunkleosteus. The genus was first described in 1873 by paleontologist John Strong Newberry as Dinichthyes torelli. The genus was renamed Dunkleosteus in 1956 by Robert Hibbard, in honor of David Dunkel of the Cleveland Museum of Natural History. Dunkleosteus's body length has been a moving target. For most of the 20th century, length was estimated at 6 to 9 meters. In 2023, paleontologist Russell Engelman published a reevaluation. He used a relationship between head and body proportions in modern fish. He concluded the actual length may have been closer to 4 meters. Body mass estimates range from 1 to 4 tons depending on the length reconstruction used. Whether 4 or 9 meters, Dunkleosteus was the apex predator of its world. The skull and pectoral region were encased in interlocking bony plates. They were fused into a rigid armor box. The mouth contained no true teeth. Instead, the plates of the upper and lower jaw bore self-sharpening occlusal surfaces. These surfaces were formed by bone-on-bone wear during feeding. As the upper and lower plates ground against each other, they remained sharp throughout life. In 2007, paleontologists Philip Anderson and Mark Westneat published a study in Biology Letters. They used computer modeling to estimate the bite force of Dunkleosteus. The result was extraordinary. The bite force was 1,500 to 4,500 newtons at the tip. This places it among the strongest bites of any vertebrate. The mouth could open in approximately 20 milliseconds. It used a 4-bar linkage system, a mechanical arrangement of bones that creates rapid suction. The mouth functions simultaneously as a vise and a vacuum. Dunkleosteus likely fed on other armored fish, ammonoids, and possibly each other. Fossil specimens have been found with regurgitated bone fragments that include other Dunkleosteus remains. Cannibalism appears to have been routine. The Cleveland Shale of northeastern Ohio represents the type locality. It has produced dozens of well-preserved skull specimens. The posterior body was unarmored. It probably resembled a shark in shape. Direct fossil evidence is sparse because cartilaginous portions rarely fossilize. Eyes were positioned forward on the head, providing binocular vision typical of an active predator. The animal lived in shallow seaways covering what is now North America during the Devonian. 6 species are currently recognized. Dunkleosteus torelli is the largest and best known. The End-Devonian Hagenberg event approximately 358 million years ago wiped out all placoderms. It ended 200 million years of armored fish dominance. The event was likely triggered by widespread ocean anoxia and sudden cooling during the Fenian-Carboniferous transition. Dunkleosteus is one of the most popular paleontology specimens in museums worldwide. Mounted skulls can be seen at the Smithsonian, the Cleveland Museum of Natural History, and the American Museum of Natural History. 9 creatures are now behind us, but the question that connects them is still ahead. 2 of the 9 creatures we have just examined came from one place. Hallucigenia and Opabinia were both Cambrian inhabitants of what is now British Columbia, Canada. Their preservation was made possible by the Burgess Shale, a black mudstone formation that was discovered by Charles Walcott in 1909. Why does the Burgess Shale preserve such weirdness so well? The answer lies in taphonomy, the study of how organisms become fossils. The Cambrian seafloor of British Columbia was at the base of an underwater cliff. Periodic mudslides buried entire ecosystems in oxygen-poor sediment. The lack of oxygen prevented decay. The fine sediment captured even soft tissue. As a result, the Burgess Shale preserves animals that other rock formations cannot capture. The Cambrian explosion itself was a 5 million year window beginning roughly 540 million years ago. During this window, most modern animal phyla first appeared. Body plans that look familiar today, including arthropods, mollusks, and chordates, all emerged in this brief geological moment. The Burgess Shale captures the experimental phase of that explosion. Many body plans appeared, persisted briefly, and disappeared. Hallucigenia and Opabinia are surviving members of that first wave of experiments. Stephen Jay Gould, in his 1989 book Wonderful Life, used these creatures to argue something profound. He said that early animal evolution produced experimental body plans that later disappeared. Most evolutionary history, in his view, was contingent. If you replayed the tape, you would get a different set of survivors. Conway Morris, the Cambridge paleontologist who reinterpreted Hallucigenia, has argued the opposite. He believes convergent evolution constrains weirdness. Similar problems produce similar solutions. Even in a replayed evolution, the same body plans would emerge. The debate between Gould's contingency and Conway Morris's convergence remains active in evolutionary biology. The role of unfilled ecological niches in driving experimentation cannot be overstated. The Cambrian seafloor was full of new ecological opportunities. There were no established competitors. There were no established predator-prey dynamics. Every body plan was potentially viable. As ecological niches stabilized, the range of viable body plans narrowed. Later periods produced fewer radically novel body plans. The genetic toolkit available to early animals was the Hox gene system and developmental flexibility. Hox genes regulate body plan formation in all bilaterian animals. Small changes in Hox gene expression can produce dramatic morphological differences. The relationship between disparity and diversity in evolutionary biology is fundamental. Disparity is the variety of body plans. Diversity is the number of species within those plans. The Cambrian had high disparity and low diversity. Modern ecosystems have low disparity and high diversity. Most weirdness disappears once a niche is locked in. But Cambrian weirdness was not the only kind evolution produced. Many later, strange body plans evolved for very different reasons. Across the 9 creatures we have just examined, 4 engines of evolutionary weirdness can be identified. Each engine produces strange morphology through different mechanisms. Sexual selection is the first engine. Walliserops' trident is the clearest example. The structure exists for male versus male combat. The largest trident wins. Diplocaulus' boomerang head may also be partly sexual. The display hypothesis remains active. Sexual selection drives the most extreme morphology because it is self-reinforcing. The trait that wins competitions is the trait that gets passed on. Runaway selection, also called Fisherian processes, can drive a trait toward absurdity within a few generations. This is why peacock tails exist. Niche specialization is the second engine. Tanystropheus' neck is a niche specialization product. The animal evolved to fish from land into deep water. The neck is the tool. Helicoprion's buzzsaw mouth is a niche specialization product. The animal evolved to slice soft prey efficiently. The whorl is the tool. Therizinosaurus' claws are a niche specialization product. The animal evolved to hook foliage in a humid Mongolian floodplain. The claws are the tool. Niche specialization produces extreme solutions when the niche is extreme. There is no advantage in moderate adaptation if the niche demands extreme adaptation. Convergent evolution is the third engine. Yiqi's membrane wing is a convergent solution. Bats evolved similar membrane wings 50 million years later. Pterosaurs had evolved similar membrane wings even earlier. 3 independent lineages reach the same solution. Hallucigenia's dorsal spines are a convergent solution. Many soft-bodied animals have evolved phosphatic spines for defense. Convergent evolution shows that some solutions to evolutionary problems are nearly inevitable. The same engineering challenges produce the same engineering answers. Ontogenetic development is the fourth engine. Diplocaulus juveniles have normal heads. The boomerang develops only in mature adults. The strangeness is built up through development, not present from the start. This pattern is common in extreme morphology. Many bizarre features develop late, when the animal has time to grow them. The role of accident, of historical contingency, also matters. Some weirdness is just an artifact of evolutionary history. The shape of the human ear, for example, reflects the migration of fish jaw bones into the mammalian middle ear over hundreds of millions of years. The result is functional but unusual. The interaction of all 4 engines in single species is common. The Dunkleosteus shearing plates combine niche specialization, ontogenetic development, and historical contingency. The mouth opens through a 4-bar linkage that is itself a contingent outcome of placoderm anatomy. Understanding which engine is at work matters for interpreting fossils. It also has implications for predicting future evolution under climate change. As ecosystems shift, new niches will open. New extreme morphologies may evolve. But our knowledge of what evolution can produce is biased by what survives in the rock record. The fossil record is incomplete. Estimates suggest fewer than 1 in 10,000 species ever fossilizes. Soft-bodied animals are nearly always lost. Cartilaginous fish like Helicoprion lose all their body's soft tissue. Only the heavily mineralized teeth survive. We do not know the body shape of Helicoprion with certainty. Even bony animals are biased by environment. Animals that lived in deep ocean settings preserve poorly. Animals that lived in upland forests preserve poorly. Animals that lived on tropical coral reefs sometimes preserve well, sometimes not, depending on local taphonomy. The exceptional fossil sites are called Lagerstätten. They preserve unusual amounts of detail. The Burgess Shale is one. The Solnhofen limestone of Germany, where Archaeopteryx was discovered, is another. The Yixian Formation of China, where Yiqi and Beipiaosaurus were discovered, is another. The Monte San Giorgio of Switzerland, where Tanystropheus was found, is another. Lagerstätten are rare. Only a handful exist for each geological period. Why we know so much about the Burgess Shale and so little about most other Cambrian environments comes down to local burial conditions. The Burgess Shale captured a single moment of a single ecosystem. We have almost nothing comparable for thousands of other Cambrian environments. The scale of what is missing is staggering. Estimates suggest millions of species have lived on Earth. Only a few 100,000 species are known from the fossil record. Entire ecosystems have left no record at all. Paleontologists call these dark biota. Soft-bodied creatures from deep-sea environments are particularly poorly known. The role of taphonomy, the study of post-mortem processes, has shaped what we see. Bone fossilizes well. Cartilage fossilizes poorly. Calcite shells fossilize well. Chitin fossilizes poorly. Phosphatic spines fossilize well. Silk-like materials fossilize rarely. The Yiqi specimen is exceptional precisely because it preserves the membrane-soft tissue. Most pterosaurs, which had similar wings, do not preserve their wings in detail. Only at sites with exceptional preservation do we see the membrane structure. The Antarctic and tropical regions are fossil record blind spots. Fieldwork is difficult. Outcrops are limited. Whole continents and time intervals are nearly invisible. Cretaceous Mongolian floodplain ecosystems, like the Nemec Formation that produced Therizinosaurus, are exceptional environments. They preserve unusually well because of the climate and depositional setting. The challenge of dating exceptional finds adds another layer. Radiometric dating requires volcanic ash layers. Ash layers are not always present. The implication of all this bias is sobering. Every weird animal we know of probably has weirder relatives we do not know about. The strangest creatures in evolutionary history may be ones we will never see. Our picture of evolutionary weirdness is limited by the geology that recorded it. The patterns we identify may reflect preservation more than they reflect biology, but the weirdness we do see has been repeatedly mishandled by the science meant to interpret it. A pattern emerges across the 9 creatures we examined. They were all misidentified, sometimes for decades, sometimes for over a century. Conway Morris reconstructed Hallucigenia upside down and backwards in 1977. The error was not corrected until 1991, when Lars Ramsgold and Hao Xianguan published the reorientation. Even then, the head and tail were misidentified for another 24 years. The full picture did not emerge until 2015. The 1972 conference audience laughed at Opabinia. Whittington had to convince his colleagues that the creature was real. Even then, his reconstruction has been refined 4 times since. The latest refinement, by Daly in 2023, addressed the eye geometry. Von Meyer mistook Tanystropheus vertebrae for pterosaur wing fingers in 1852. The correction did not arrive until 34 years later. In 1886, Malieve classified Therizinosaurus as a turtle relative in 1954. The correction took decades to emerge as additional skeletal material was discovered. The full classification as a theropod dinosaur was not established until the 1980s. Karpinski placed the Helicoprion whorl on the upper jaw, curling outward like a forehead horn. In 1899, the correction did not arrive until 2013. 114 years separated the original misidentification from the CT scan base resolution. Walcott classified Opabinia as a crustacean in 1912. The classification was not corrected until 1972. 60 years passed before the laughter could begin. The pattern is striking. When an animal's anatomy does not fit known forms, scientists default to the closest familiar match. Trilobites are familiar. Pterosaurs are familiar. Turtles are familiar. Sharks are familiar. The error is not careless. It is the natural product of cognition. Faced with the unknown, the brain reaches for the known. The role of preconception in shaping interpretation is profound. Even the original specimens have been described as deliberately mounted upside down by Conway Morris because the orientation seemed obvious to him. Modern tools have changed this. CT scanning, finite element analysis, and morphometrics resolve cases that visual inspection cannot. The Tanystropheus species distinction was resolved by CT scan. The Helicoprion whorl placement was resolved by CT scan. The Walliserops trident function was resolved by morphometric comparison to beetles. Why these errors took decades to correct is partly publication and prestige. A scientist who has built a career on a particular interpretation has incentives to defend it. Stephen Jay Gould's role in publicizing the Cambrian reinterpretations gave them broader currency. Without his coverage, the corrections might have remained inside paleontology. The lesson is that extreme weirdness should be expected to be mistaken. The continuing frontier suggests current reconstructions might also be wrong. Today's confident interpretation may be tomorrow's embarrassing error. But strange creatures from the deep past are not entirely separate from us. Their lineages continue through their descendants. Hallucigenia's lineage continues. Modern velvet worms in the phylum Onychophora are the closest living relatives. They live in tropical leaf litter today. They squirt sticky slime to capture insects. They retain the soft, fleshy, unjointed legs that Hallucigenia possessed. The body plan persists, just downsized. Opabinia's contribution to the modern arthropod ancestry is genetic, not morphological. The five-eye configuration disappeared. The grasping proboscis disappeared. But Opabinia's developmental innovations contributed to the lineage that became insects, crustaceans, and arachnids. Modern arthropods are descendants of stem group animals like Opabinia. The five-eye design vanished because it was outcompeted by jointed limb predators. Helicoprion's relatives include modern chimeras and silver sharks. These cartilaginous fish retain elements of the eugeneodontid lineage that Helicoprion belonged to. None have buzzsaw mouths. The slicing spiral strategy disappeared with the end Permian extinction, but the broader cartilaginous fish lineage has survived. Therizinosaurus's lineage continues through birds. Theropods are the ancestors of all modern birds. Therizinosaurus itself left no direct descendants, but the broader theropod radiation produced over 10,000 bird species alive today. Modern parallels for Tanystropheus' neck exist in the form of giraffes. The giraffe has one of the longest necks of any modern animal, but the giraffe achieves this length with 7 cervical vertebrae, the same number as humans. Tanystropheus had 13 vertebrae. The strategies are different. Brachiosaurus and other sauropods had similar absurdly long necks but achieved them differently. Diplocaulus has no modern analog. The boomerang head appears to have been a one-time experiment. No living amphibian has comparable skull morphology. The ecological role of an ambush predator with a wide head lying on the riverbed and striking upward has not been recreated. Walliserops' descendants are absent. There are no living trilobites. The lineage was wiped out by the end Permian extinction. But the horn weapon strategy continues. Modern beetles use exactly the same kind of structure for the same kind of male versus male combat. The form has been recreated by an entirely different lineage. Dunkleosteus' complete lineage extinction is a striking case. Placoderms left no descendants whatsoever. The end Devonian Hagenberg event wiped out the entire class. 200 million years of armored fish dominance ended in a few million years of mass extinction. The role of the Hagenberg in cleaning out armored fish reshaped subsequent fish evolution. Cartilaginous fish and bony fish filled the empty niches. Modern strange animals exist all around us. The platypus combines mammalian, reptilian, and bird-like features. The proboscis monkey has a nose that defies explanation. The okapi is a giraffe relative that looks like a zebra. Modern weirdness tends to be mosaic rather than experimental. The thread of evolutionary continuity across mass extinctions is unbroken in some lineages and severed in others. The strange creatures of today are evidence the experimental phase never fully ended. 9 creatures. Each one looked too strange to be real. Each one was. Walliserops carried a literal trident on its face. Yi Chi flew on bat wings. Diplocaulus wore an anvil for a head. Hallucigenia walked on stilts with tentacles waving. Opabinia probed the seafloor with 5 eyes and a vacuum hose. Tanystropheus extended a neck 3 times longer than its body. Helicoprion sliced prey with a buzzsaw lower jaw. Therizinosaurus picked leaves with meter-long scimitar claws. Dunkleosteus bit through armored fish with self-sharpening shearing plates. The recurring theme is that life produces forms that defy expectation. Paleontologists have repeatedly failed to predict what fossils would show. Almost every weird animal in this video was misidentified at first contact. The lesson is that looks impossible is not the same as is impossible. Evolution does not respect the boundaries of human intuition. It produces what the genome and the environment together permit. The Cambrian explosion is proof of evolutionary plasticity. In a 5 million year window, body plans appear that have no modern equivalent. The plasticity has not vanished. It has been constrained, but not eliminated. The Devonian and Triassic produced new weirdness of different kinds. After the Cambrian close, the Cretaceous produced Therizinosaurus. The late Pleistocene produced Megaloceros, the Irish elk with antlers spanning nearly 4 meters. Strange anatomy keeps emerging. The implication for current biodiversity is that strange animals exist now. We just do not notice them because they live in places we do not look. The deep sea, the soil, the leaf litter, the abyssal plain, these contain creatures whose body plans would shock us if we could observe them clearly. The implication for life on other planets is that the universal mechanisms producing weirdness here may produce weirdness elsewhere. Sexual selection, niche specialization, convergent evolution, and ontogenetic development are not Earth-specific. The role of mass extinctions in cleaning out weird body plans is double-edged. Extinctions destroy strange forms. They also open ecological space for new strange forms to emerge. The cycle continues. Why some weirdness persists and other forms vanish has more to do with luck and environmental change than with evolutionary fitness in any absolute sense.